Uh, Good morning, everyone, and thank you for joining us this morning for our first AMM Science and Technology Committee's webinar series. Our first webinar going to be about RNA modification. Appreciate West Coast time getting up early to see that, and we have a good lineup for you to go over the aspects of the function of mRNA, modified mRNA, how to characterize it, and so forth. Next slide, please. So the agenda for today is welcome and logistic, little background on what is AMM, and then the three topics we have three speakers. We're going to go through RNA methylation in medicine, and the mRNA modification, their cellular biology, therapeutic use, and clinical impact. And lastly, identification and characterization of mRNA modification using LCMS. And we're going to end it with a Q&A. Throughout the presentations, we would ask everyone to hold off in terms of your questions and put them in the Q&A box. That way, all the questions will be addressed to the speakers, and we can have a more of a holistic answer. Next slide, please. Again, just in terms of general, this webinar will be recorded and will be available later for view and to go over it again. And again, questions, please enter your questions in the Q&A box, not in the chat. And if we'll try to get to all the questions. If not, we'll see if we can do something in terms of address them after the webinar. Next slide. So today, my name is Khaled Yammoud, and I'm the co-chair of the AMM Science and Technology Committee, and I'm going to be moderating the sessions. We have three speakers. We start with Chang Hu, who's from the University of Chicago, followed by Tim Mercer from University of Queens, and Mark Dickman from University of Sheffield will go over the characterization of mRNA, modified mRNA. Next slide. So a little bit about AMM. So AMM was established in November 2023 by the mRNA community to advance its priorities. This way we can have a message, to advance the mRNA and show how good it is in terms of application to different therapeutics. Next slide. So one of our, this is what we do. You can take a look at that once it's available to look. But in general, one of the properties of AMM is to bring together the community to amplify and discuss scientific progress, which we are going to do today. Just focusing on the science, and we're hoping that we'll keep these webinars coming. That way, people keep getting educated from the scientific standpoint regarding mRNA. Next slide. So AMM now has over 60 organizations that have joined from around the globe, with members from across the community. This includes biotech, pharmas, academics, CDMOs, and service providers. And AMM has been engaging with the regulatory to see how they can help facilitate the advancement of the drugs and the requirement for CMC. Next slide. So as you can see in this here, it's actually global. We have representatives from all over the world. Next slide, please. And this is kind of a timeline in terms of committees, and one of the priorities here is the Science and Technology Committee. So we started this webinar series, and the goal is to have a forum for discussion of the critical scientific topics. We'll hold one webinar every 6 weeks or so. So the next one will be around December. And also, feel free to email us if you have any ideas. And the hope is by spring and summer 2025, we will conclude this webinar series of our first AMM scientific meeting in person, to also to have some presenters and keep building up on that. Next slide, please. And one more. So lastly, before we go into our to our speakers to cover the topics that on the agenda, November 13 to 15, AMM is holding its Ascend meeting. It's open to non-members. We're bringing together leaders from across the country to discuss the latest on science, investment, development, manufacturing, and delivery as related to mRNA. So it'll be great to see you there, and you can go to the website and sign up, and hopefully we can see you in that meeting. Next slide. So next, I would like to we go through our first speaker, who's Hu Chang from University of Chicago, talking about mRNA methylation. Professor Chang, he is currently the professor at the Department of Chemistry and Department of Biochemistry and Molecular Biology at University of Chicago. His research spans a broad range of chemical biology, RNA biology, and genomics. His recent research concerned reversible RNA and DNA methylation and biological regulation. His research group discovered the first RNA demethylase and shows that reversible RNA methylation significantly affect gene expression regulation. Chang, please take the floor is yours.
Thank you. Can you hear me?
Yes.
Yeah, great. Thanks for this opportunity. Yeah, it's truly a pleasure to have this chance to share with you some of our research. Can we advance slide to the next one? Yeah, I started this with thinking about the simplicity of the genome. As we probably know, our genome is about 3 billion base pairs long, but each of us contains tens of trillions of cells. And for that 3 billion base pair genome, only 1.5% encoding real protein sequence. So the question's been, what sort of give the complexity of human? It can't be just the sequence of the genome. Next, please. It turns out on the genome, there are these chemical modifications. On the DNA, there are these methylations, but there's also nucleosomes. These are proteins that package DNA together. And at the tail of these nucleosomes or histone tails, there are also chemical modifications. Next. So in a way, our genome is just not a flat information context. Instead, it has these modifications that give different patterns. Next. And these methylation could be recognized by binding proteins that will regulate transcription. So in a way, next please, you'll see this is almost like a key. It's not a flat key. It has these teeth, the different patterns or so-called epigenetics. These patterns are very different for each segment of DNA for different cell types. So at promoters or enhancer sites, you have different modification patterns and you have different ways to turn on and off a gene. Next, please. So this is well known in the central dogma. You go from DNA to mRNA to protein, and there are modifications on DNA and histones that encodes vast amount of complexity. So we asked back in 2010, next please, whether mRNA carries similar sort of modifications that could broadly regulate genetic information flow in the central dogma. Next, please. In 2011, we indeed discovered that modifications on mRNA could be reversible in a way that means you can shape the different patterns on the same mRNA molecule by adding or removing methyl groups. Next, please. And this modifications called N6-methyladenosine or M6A is the most abundant mRNA modification in mammalian systems. In general, a mammalian messenger RNA molecule will carry, in average, about 3 internal M6A. And this is conserved in most eukaryotes and essential in mammals. So this was back discovered in 1970s, but because of lack of tools or lack of methods, the exact effect of this methylation was not so clear. Next, please. So next, please. We discovered 2 demethylases that could reverse this methylation or remove this methylation. Next, we and others have also characterized the big methyltransferase complex. This is a 1000 KD complex that install the methylation. Next. Over the last decade, my laboratory and other labs have really worked on the binding proteins. So it turns out hundreds to thousands of mRNA could be M6A methylated, and they can be recognized by different binding proteins. These binding proteins binds to methylated RNA, regulate their stability, translation, and cellular localization. Now, these effects are quite dramatic. Next, it affects all kinds of physiological outcomes. I'm going to actually introduce a little bit on cell therapy, but there also next affects the cancer therapy. Next, immunotherapy, I'm going to touch upon a little bit. And the next, please, and neurodegeneration. So let's start with cell therapy. Next. I want to give you an example of one of the proteins, the YTHDF2 binds methylated RNA and mediates decay of methylated RNA. Next. So previously, we've shown that this protein plays critical roles during stem cell differentiation and early development in mammals. Here's one example. When fertilized egg started to develop, these maternal mRNAs that needs to be decayed in order for zygotic state to take over. The decay of these maternal messenger RNAs is partially mediated through methylation and YTHDF2. Next. Another good example is this collaborative work with two laboratories shown down there. They were studying neural stem cell differentiation. You go from a neural stem cell all the way to final neurons. Upon that differentiation, you see this methylated RNA comes and go. This methylated RNA help drive translation in order to drive differentiation, but they also need to be decayed in order for cell to move their cell state, eventually reaching to the final neuron state. Now, without methylation or without YTHDF2, this differentiation will be partially blocked. You'll be stuck, and you won't be able to have final differentiated neurons. So next slide. In a way, essentially what we view this as shown in this slide, if you go from a stem cell to a progenitor cell to the final cell state, each cell state have characteristic mRNA patterns, right? These are the mRNAs dictates that specific cell state. If you want to move on from stem cell to progenitor cell to final cell, you not only need to have new transcripts that represent the next cell state, but you also need to get rid of transcripts that dictates the previous cell state. Now that sort of a clearance of previous mRNA is actually done by methylation and through YTHDF2 that decays those RNAs, so drive cells to differentiate into next state. Next, please. So we decided to look into this using a well-studied system, hematopoiesis. Here you have stem cell differentiated to progenitor cell, and then you differentiate into different cell lineage. Eventually, you get all kinds of immune cells and blood cells. Next, please. So we postulated that, again, the mechanism I just talked about would play a critical role to regulate the stem cell progenitor cell self-renewal versus differentiation, right? It either self-renews or differentiates. And we believe the gate is the pathway I just shared with you. The YTHDF2 will needs to clear messenger RNA from stem cells, progenitor cells, otherwise these cells cannot be differentiated. So we wondered if we block YTHDF2, could we drive stem cell self-renewal so we can expand these stem cells for therapeutic applications. Next, please. There's obvious clinical needs. For instance, bone marrow transplant. As we all know, patients have leukemia or other diseases would require bone marrow transplant. And the critical limiting step, next slide, is really to identify donors that have the key antigens matched to the recipients. That is a major challenge, right? Even if you find the correct match, about 30–40% of transplant cases would have immune complexities, and the patients will suffer for long-term immune response, as a so-called GVHD. Now, there's another way to solve this challenge. That's use a cord blood stem cell. Now, these stem cells require much less stringent antigen matching, and they typically prevent GVHD, so you won't have the immune complexity. So far, this cord blood stem cell transplant only accounts for 20% of US transplants. Next, please. The major problems being the lack of materials. So for adult recipients, 2 cords are required for 1 adult. So there's just not enough typically cord blood stem cells available for this transplant. You have to match them, and you also have to have sufficient quantity. Next, please. For this reason, previously Professor Linheng Li came talk to me, and we decided to have a test. We wondered if we block YTHDF2, could we drive self-renewal of these cord blood stem cells? And that way we could expand these stem cells to many folds and would have sufficient materials for transplant. Next, please. So we tested this in human cord blood hematopoietic stem cell. As you can see, if we knock down YTHDF2, that block sort of the decay of methylated messenger RNA, we can expand human cord blood stem cell about 14 folds after 7 days. Next. And we observed that these stem cell markers are elevated. So we not only expand them, but we also increase the stemness of these stem cells. Next, please. And Linheng's lab also did in vivo experiments. Next, please. They actually took 1:1 ratio of control cell versus the expanded stem cell injected into mouse bone marrow. In longer term, they learned that actually, even if we perform the 1:1 ratio reinjection, inside of the animal, we saw additional 4 to 8 folds expansion. So not only ex vivo, we obtained 14-fold expansion. These stem cells expanded by YTHDF2 knockdown have increased the stemness. And once they got into the animal bone marrow, we got additional expansion. So this is actually an ideal case. We not only prepared sufficient amount of stem cell, but we also dramatically increased their stemness and their ability to further expand inside bone marrow. Next, please. So this would be an ideal case if we can implement into clinical for transplant experiments, but there are other possibilities. For instance, one can start to think about inhibiting YTHDF2 to expand mesenchymal stem cells for other cell therapies. One example, next please, is cartilage repair, right? When we get older, we have these knee problems, and we wondered, could we also expand mesenchymal stem cells for tissue repair and tissue regeneration. These are areas would be very interested to explore in the future. Next, please. So I promised I'm going to give I was going to give 2 examples, stem cell therapy being one, and another one is immunotherapy. As we all know, immunotherapy has been revolutionary in treating cancer patients. These are the so-called PD-1, PD-L1 that inhibits T cell response to the presence of tumor cell. Now, only a only a fraction of patients that are sort of responsive to immunotherapy. In many cases, the T cells just can't have a chance to gain access to tumor. Next, please. So it turns out when we talk about immunotherapy, it's not just simply T cells interacting with tumor, but there's also this tumor microenvironment. Many of this tumor microenvironment prevents T cell access to tumor. So if one can sort of modulate this tumor microenvironment, allow T cell to access tumor, this would dramatically open the sort of the scope of immunotherapy. Next, please. During our study, we realized the protein I described to you, YTGDF2, play a critical role to promote tumor microenvironment that suppress immune response. Next, please. In addition to YTGDF2, there's also YTGDF1. So these collectively play critical roles to sort of help establish this immune barrier that prevents immune response to tumor. Next. So in collaboration with my colleague, Dr. Rolf Wassenbaum, the chair of Radiation Oncology Department at the University of Chicago, we decided to look into this. Next, please. And Rolf realized when he applied radiotherapy to patients, there are always a group of patients not responsive to radiotherapy. And he looked carefully, realized the elevation of the gene YTGDF2. This is consistent with our hypothesis. This gene plays a critical role to establish immune barrier. Next, please. We move to model experiments. In mouse, we realized whenever we knock down YTGDF2, we can substantially sensitize radiotherapy, and together with radiotherapy and immunotherapy, we can almost eradicate tumor. Next, please. We also developed the small molecule inhibitor. This is not ready for clinical application. This is really a tool compound, but when we apply this molecule that inhibits YTGDF2, as you can see down here, in mouse model, when we combine with radiotherapy and immunotherapy, we have a very dramatic effect in inhibiting tumor growth. Next, please. Next, please. In collaboration with my other colleague, we look at the colon cancer. These are real human samples. As you can see, the presence of the other protein, YTGDF1, that also regulates mRNA, methylated mRNA. We saw an inverse correlation with immune infiltration or T cell infiltration. The higher YTGDF1, the less T cells in the tumor surrounding areas, again indicating this is another pathway to sort of set up immune barriers. Next, please. So in mouse experiments, if we knock down YTGDF1, we can increase response to immunotherapy from 40% all the way to 100%. Next, please. We also developed the small molecule inhibitor as a tool compound. Again, when we apply this in combination with anti-PD-L1 immunotherapy, we saw very promising effects. So far, I showed you that these are very good targets to enhance radio or immunotherapy. We indeed are going to have a small biotech develop these inhibitors, and hopefully this will go on to clinical trials and eventually hopefully will impact healthcare, particularly for cancer patients. Next, please. So what I just introduced is actually regulation from mRNA to protein, right? What I introduced are the modifications on mRNA that impacts mRNA stability translation. Next, please. I know this is the mRNA sort of a community, but I'd like to actually spend the next 5 minutes and give you sort of a flavor of the other side of the coin, that the RNA methylation also impacts transcription. That is from DNA to mRNA. Next, please. So while we were working on mRNA methylation, next, please, we realized that the methylation also occurs to non-coding RNAs on the chromatin. Next, please. When we deplete these methyl groups by knocking down methyltransferase, we saw dramatic chromatin state changes. As you can see, that the entire chromatin state become more open, and we started to speculate that this is the transcriptional regulation. Next. What we showed in 2020 that there are these non-coding RNAs that really help assemble transcription active complex, and these RNAs themselves are regulated through methylation. So if they're methylated, they could be recognized by the binding proteins that recruit additional machineries to either decay this RNA or add histone marks to suppress transcription. Next, please. So a year later, 3 laboratories independently made very similar observations that these methyl groups on chromatin-associated RNA can indeed impact transcription at the local chromatin state. Next. So all of us independently showed that there are these non-coding RNAs on the chromatin that can be methylated. Next, and the methyl group could be recognized by binding proteins, and this shape the local chromatin state and transcription. Next, we've also discovered additional binding proteins that recognize chromatin-associated RNA methylation to impact transcription. Next, and we also found proteins that remove methyl group, so that can reverse the regulatory process. Next. Now, this has a dramatic effect. For instance, if we take the demethylase we discovered and overexpress them in the plants, and in rice, we saw about 30 to 50% increase of their yield and biomass, and not only in rice, next, but also potato. And what we realize is that methylation and demethylation dramatically tune the chromatin state of stem cells in plant, and that regulates plant development and plant growth. Next. Now, we borrowed that message and decided to look into mammalian systems. So essentially, what we realize when we talk about chromatin state regulation, we not only need to think about DNA modification, histone modifications next, we also need to think about this nascent RNA that could be modified. The chemical group could attract binding proteins, attract additional machineries to modulate local chromatin state. So what I showed here is the M6A as one example. Next, please. We asked whether there are additional groups that could also occur on this nascent RNA and modulate chromatin state and transcription. Next. So we turn to cytosine methylation. It turns out there's this tumor suppressor TAT2, which was thought to be a demethylase of DNA cytosine methylation. Now, this then is frequently mutated in human leukemia. Next. There's also this IDH, the metabolic enzymes frequently mutated as well, and their mutation produce the oncometabolite 2-hydroxyglutarate, which actually inhibits TAT2 that contributes to initiation of tumor. So IDH mutant occurs widely in human leukemia as well. Next, but also in 75% of low-grade glioma and in other solid tumor. Additionally, TAT2 levels substantially downregulated in human cancers such as glioblastoma, prostate cancer, melanoma, and even breast cancer. So TAT2 is a well-known tumor suppressor. It's either frequently mutated or substantially downregulated. Next. What we found is a chromatin regulation pathway through RNA M5C. Essentially, TAT2 serves as a gene suppressor. The loss of TAT2 activates gene expression with the pathway revealed by us, shown on this slide. When we knock down this pathway in the Y-type human leukemia, you see on the left, we have some inhibition of leukemia proliferation, but when you have a TAT2 mutant leukemia, which is the right last panel, we got synthetic lethal, meaning that with TAT2 mutation, the pathway we discovered is upregulated, and the leukemia cells rely on this pathway to proliferate. If we shut down this pathway, we selectively eliminate these groups of leukemia cells. Next, so this is sort of the most recent results we obtained, not only revealed the clinical importance of RNA methylation that regulates chromatin state, but also identified the targets that we can develop future therapies to precisely target a subclass of leukemia, but also other human diseases caused by TAT2 mutation. Next. So that's sort of my my general summary. I hope I sort of give you a flavor of mRNA modification that could impact medicine, but also chromatin RNA methylation that have promises to develop new therapeutics. Thank you very much.
Thank you, Dr. He. Appreciate that. So next we will be moving to our next speaker, Timothy Mercer from University of Queens, and he's the director of BASE. Timothy is a group leader at the Australian Institute for Bioengineering and Nanotechnology at the University of Queens, and he is the scientific director of BASE Nucleic Acid Synthesis Facility. His group, he also leads a diverse laboratory and bioinformatic research group into the expression and splicing of synthetic genes. Tim, please go ahead, and the floor is yours.
Yeah, fantastic. Thanks for the introduction, Colin. Yeah, so look, so my name's Timothy Mercer from the University of Queensland and also the base facility. Maybe progress onto the first slide, please. Just before we begin, just as a quick disclaimer that myself and also my research lab do sort of, I guess, benefit from research funding from the following commercial organizations. And obviously, these views and opinions that are expressed are my own as well. So please move on to the next slide. Oh, can you go forward? Right, it seems to have the text. Okay, cool. Right, maybe it's just that slide. So maybe if you go back one slide, apologies. It looks like there's a little bit of a. That's right. It's just an introductory slide, just to quickly sort of introduce myself. As Colin was saying, so my name's Timothy Mercer from the University of Queensland. And there I lead what we call the base facility. So this is a national Australian facility that really focuses on mRNA research and manufacture. And it's in total, it's probably a group of about, there's about 30 scientists, and we're together kind of, I guess, focused on the development of new mRNA medicines. So that includes different aspects of drug development, so mRNA design, but also manufacture formulation, candidate screening, and a lot of sort of preclinical evaluation and testing as well. And I think if there's kind of one thing which kind of joins the group together, the scientists together, it's that kind of, I guess, that sort of our focus is really trying to, I guess, understand the mechanisms of action for this kind of, I guess, the next generation of sort of mRNA medicines. And so I was invited today to talk by the AMM committee on about how we use RNA modifications, how we use them both within our clinical development and also our manufacture and also our analytics as well. And I mean, whilst trying to provide us like a really good insight into, I guess, the kind of the biology, the sort of how the cell uses RNA modifications to kind of control its expression and regulate those cellular processes, I guess my remit, or what I was told from Colin, was to kind of, I guess I'll provide a little bit more of an overview, a bit more of a review of kind of RNA modifications in terms of, in particular, from a sort of a clinical or a therapeutic development perspective, and obviously with that particular focus on sort of mRNA medicine, so mRNA vaccines and therapies. So with that brief introduction, maybe to the next slide, please. Okay, cool. So look, I mean, let's just start with the RNA modifications themselves. I mean, I think the story of RNA modifications is really probably about as old as life itself. It probably began on, at the dawn of life about 4.5 billion years ago. And that's because pretty much our most ancient genes, so the genes, the ribosomal RNAs, the tRNAs, the genes that are shared across all domains of life are all pretty intensively modified. And these modifications are really required to ensure the folding and the actual correct function of these RNAs. And so you can imagine there's a lot of RNAs within the cell that are modified. tRNAs, ribosomal RNAs, a whole class of smaller structural non-coding RNAs as well, and obviously the mRNAs themselves. So there's a huge diversity and also abundance of RNA modifications in there. And we're still also discovering new. I mean, I thought it was kind of interesting. There was a recent report on glycosylated RNAs, so RNAs that have got glycan residues conjugated that are kind of presented on the outside of that cellular membrane. So the short story is there's a massive diversity. I mean, I think there's like hundreds of RNA modifications within the cell, and there's still more being discovered as well. And then there's also, to complement those, in some ways, those natural endogenous modifications, there's also a whole array of synthetic or artificial modifications as well. Next slide, please. Now, if we kind of, I guess, focus just specifically on the mRNA, as Chan said, there is like that kind of a diversity, again, of those modifications. And again, these modifications, which really extend across the full length of the mRNA, are added dynamically by that sort of network of enzymes. We saw a couple of examples there, you know, these kind of, there's this network of, I guess, enzymes, these protein networks that essentially are able to, in the case of like writer enzymes, add modifications to the mRNAs. They can read or interpret the modifications, so reader enzymes, or alternatively, you know, remove or delete those enzymes, delete those modifications, so eraser enzymes. I mean, the reality is it's a bit more complex than these three simple buckets. But the reality is there is this kind of this network of proteins that's able to modulate, and on a global level, as you just saw, on a global level, modulate the modifications across the transcriptome. And this has led to this idea of like the epitranscriptome, analogous to the epigenome, and this idea that, you know, there's kind of this combinatorial, this sort of nucleotide code, this code of nucleotide modifications that can quite rapidly sort of modulate and regulate the translation and the stability of the mRNAs. So whilst not the topic of the talk, I'm going to focus a little bit on how those modifications have been in some ways leveraged or developed for clinical applications. But I did feel, even though this isn't the topic of the talk, I did feel I'd be a bit remiss if I didn't highlight, when it comes to RNA modifications or the clinical development of RNA modifications, you know, there is a host of actual RNA editors. Trying to just put up some examples, but I think there's a pretty wide host of RNA editors that are actually undergoing pretty active clinical development themselves. So the idea behind these RNA editors is they can essentially correct or enhance or silence gene expression in those diseased tissues. And I think there's actually pretty encouraging results, like very early stage. A lot of these things are very early stage, but early stage encouraging results from a phase 1 clinical trial for, I think, Wave Sciences, for an ADAR RNA editor as well. So an editor that's able to convert those adenine to those inosine residues. Next slide, please. So what I'm going to do then is those are all the sort of the endogenous modifications, but what I want to do is I want to kind of walk a little bit through the mRNA itself, point out the key modifications and how they've been sort of leveraged and how we use them in mRNA manufacture and sort of their role in mRNA vaccines. And the obvious place to really start is obviously at that 5' cap. This is where you know, translation initiates, where it begins. And eukaryotic mRNAs have a modified 5' nucleotide, a 5' cap. It's this modified like sort of guanine nucleotide, and it's kind of actually linked to the mRNA by this unique sort of back to front 5' triphosphate linkage. Now, in addition to that actual 5' cap, the next two nucleotides, so the first and the second nucleotide, like sort of the proximal nucleotides, can also have epigenetic marks as well. So if the first one's methylated, it's a cap1 structure. The second one, if that's methylated, we consider it like a cap2 structure. You know, those cap1 structures, they're pretty prevalent. They're pretty standard within sort of high eukaryotes, whereas the cap2 structures are more common, say, in mammals, more prevalent in mammals. But again, these are kind of these epigenetic marks that have, in some senses, also interact pretty closely with the innate immune response with those cytoplasmic sensors, and I'll touch on them in a second. But in some senses, these different epigenetic marks have kind of co-evolved with the different cellular innate immune systems to kind of start recognizing and distinguishing self from foreign RNAs. And I'm going to touch on one of those key roles in a second. But before I do, one thing the cap is critical for is really the assembly of the translation initiation machinery. You know, it kind of obviously recruits that elongation initiation factor, starts to build that machinery around it. Interestingly, it also is thought to basically interact with the protein binding, the poly-A tail binding proteins at the other end of the mRNA and sort of circularize the mRNA. So this is this kind of model of mRNA translation. And the cap obviously has a pretty catalytic sort of fundamental role in sort of catalyzing that assembly. Now, like I said, if you've got mRNA that are uncapped, they don't have this RNA modification at the end. That's actually a pretty potent signal for the cellular innate immune response to essentially detect it and kick off an inflammatory response. And so this is kind of, there's the sensors, notably things like RIG1 that are within the cytoplasm that can essentially detect those uncapped mRNAs. And for that reason, it's obviously critical when you're developing an mRNA vaccine, when you're manufacturing an mRNA therapy, that most of your mRNA molecules are capped. And it's not just our molecules, it's not just the vaccines that we want to cap. It's also RNA viruses. have got the same problem. You know, they've got a lot of the same similar solutions, a lot of similar workarounds. You know, we talk about a lot of the diversity that we see within RNA modifications within mRNA, but in the viral world, in the viral genomes, there's a massive diversity of weird and wonderful modifications, and that's also at their 5' cap. They try and cap their RNA genome to evade detection by the innate immune system. And I think, to give you an example, I think SARS-CoV-2, don't quote me on it, but I think it has, yeah, it's got like a 2O-methyl cap at the 5' end. A lot of these viruses, they can either co-opt the kind of the capping enzymes of the endogenous cell, or they have their own enzymes. And notably, the reason this is actually relevant is, like many things, we've actually kind of leveraged or engineered those viral capping enzymes in a lot of our own mRNA manufacture. So a lot of the capping enzymes that we now use in mRNA manufacture have initially been derived from those, you know, those highly efficient and often highly promiscuous sort of viral capping enzymes. Next slide, please. Oh, I keep pressing myself. So like I said, so the capping is mediated by this complex. It does happen at that transcription initiation step pretty early, you know, 20, 30 nucleotides into transcription initiation, and the capping complex is already capping the mRNA. Like I said, we can use those enzymes as well during kind of in vitro transcription in terms of the in vitro mRNA manufacture. And there is quite a few, there is a fair bit of active research in this field, looking at different capping enzymes, looking at different cap structures, and sort of actively developing them for their sort of, I guess, their clinical, their therapeutic potential. Having said that, I should say as well, when it comes to making mRNA, it's actually like a sort of like more recently, sort of co-transcriptional methods have been sort of increasingly adopted. And the reason I bring this method up is it is pretty relevant, not just to the sort of the co-transcriptional capping, the mRNA manufacture, but I also think to sort of research in RNA modifications. And that's because it does use a pretty nice method. In what it does is it basically incorporates like a synthetic 5' cap analog during transcription initiation. So what it does is you have like almost like a really small primer, an oligonucleotide primer. It might just be a couple of nucleotides that's basically conjugated onto your cap. And what you're doing is you're basically initiating transcription initiation from that sort of synthetic cap attached to the primer. So from that primer, you're initiating transcription initiation. And so it's a really efficient way of capping the mRNA. It's single pot, it's very efficient, it's very effective, you know, high efficiency. So obviously it's gained a lot of adoption within the sort of the manufacture field. But I think it's a pretty generalizable method. So you can actually add other modifications, other ligations, other conjugates to your mRNA using a similar approach, both at the 5' end and also internally within the mRNA as well. All right, so if you just, next slide, please. Just keeping an eye on time. All right, cool. So like I said, I mean, look, there's a wide range of modifications that have been sort of evaluated. So in this case, when you do the manufacture, sort of substituting out canonical nucleotides and substituting in modified nucleotides. And there's been quite a few of these different modifications, both natural and synthetic, that have been evaluated for their potential. I'm not going to go through them in too much detail at the moment. I mean, obviously, there's the M6A modification that Chuan was describing, and all the sort of, I guess, the complexity that it has in terms of actually sort of regulating sort of mRNA expression. There's also, in particular, I think 5-methylcytosine has also been shown to really improve, notably like kind of the immunogenicity or the reactogenicity. You know, the ability of that mRNA to incite the immune response. Typically, you want to try and suppress the innate immune response. You don't want to incite it. And so there's been a lot of work in trying to like look at different modifications that are sort of don't incite that innate immune response. I think though, having said that, I mean, while there's been a fair bit of sort of, I guess, research into these different aspects, and there's a fair bit of preclinical evaluation of different modes, I think it's probably actually, if you go to the next slide, please. Yeah, it's really the pseudouridine though, which is really the standout candidate when it comes to, I guess, the sort of clinical development of RNA modifications, particularly within mRNA. So this is a naturally occurring RNA modification. It helps evade immune recognition by the toll receptors, but also it notably leads to kind of also in some cases like higher stability. So it does seem to like confer a bit of sort of protection or nuclease protection from nuclease digestion and so forth. And interesting as well, it does seem to sort of support additional ribosome loading, additional sort of faster ribosome translation initiation and loading. So improving the actual expression and the translation of the actual mRNA itself. Having said that, there might be a couple of drawbacks to this, particularly some things like the ribosome loading. There was a recent paper which interestingly showed that, you know, if you have multiple pseudouridine residues within the coding sequence, it can lead to sort of frame shifting of the ribosome. So sort of frame shifting of the ribosome, which leads to those frame shift mutations, and obviously abolish the sort of the encoded protein sequence. So, I mean, look, this is, like I said, by far and away the most clinically developed, the most sort of developed mRNA. It's used in the commercially available Pfizer BioNTech mRNA vaccines and also the Moderna mRNA vaccines. Next slide, please. So it is typically used. I think it's gone. Oh, there's been a bit of with the formatting, a bit of changes, but that's all good. So in particular, it's that N1-methyl pseudouridine derivative that's currently routinely used within those mRNA vaccines. There was actually a little bit of a, I guess, a debate at the beginning of like the contribution, like the relative importance of the nucleotide modifications, the RNA modifications, to the performance of those mRNA vaccines. There was a bit of a debate, but I think, you know, it was, I mean, the lack of pseudouridine, I think, was kind of a belief, at least to be a contributing factor to some of the more disappointing like clinical results from the CureVac COVID-19 mRNA vaccine, which was unmodified. And so, you know, while there's kind of this initial debate, I think it's pretty widely considered now that the inclusion of these modified nucleotides, N1 pseudomethyluridine, have been really key to enabling the effectiveness of mRNA vaccines. And like I said, you don't have to just take my word for it. The Nobel Prize Committee seem to agree, and they obviously recognize the contributions, the research contributions of Katlin Karikó and Drew Weissman for their work in terms of looking at nucleoside modifications and the importance of those modifications in the actual sort of development of effective vaccines. And they were obviously recognized with the Nobel Prize last year. All right, next slide. I'm just keeping a bit of an eye on the time. All right, I'll quickly run through this as well. This is also, I mean, look, I think at one sense we've looked at some of the new technologies that are sort of coming along in terms of RNA modifications that are starting to kind of move their way from preclinical and hopefully into sort of the clinical research. One of the other things that we're starting to see as well is a lot of the sort of the analytical, the manufactured tools that are developed for sort of mRNA vaccines and therapies are also evolving and kind of becoming a little bit more specific for mRNA. And I want to just take a bit of a time. I mean, one of the reasons I think Khaled invited me to talk today was some of the work that we've been doing, in particular using nanopore sequencing to sort of analyze, detect, and sort of, I guess, conduct our research on these RNA modifications. And so this is kind of what we use, one of the tools in which we use to basically characterize those RNA modifications. Just as a way of a bit of a background, these kind of next generation sequencing technologies, you know, they've been extensively used for RNA sequencing now for a couple of decades. And so they've got some pretty cool technologies in terms of, pretty powerful technologies in being able to basically determine what the mRNA sequence is and quite a lot of features of it. Now, what we use is this nanopore sequencing technology. It's been commercialized by a company out of the United Kingdom called Oxford Nanopore Technologies. You can see it there on the left-hand side. It's a nanopore channel. And what it does is as you pull the mRNA or the DNA strand through that nanopore channel, what it actually does is it interferes with the current, it interferes with the ionic flow through that channel, and you can actually just measure the electronic signal. So if you're looking there in the middle in that sort of computer screen, that's what the electric signals typically look like. And we can essentially deconvolute them to identify what nucleotide is in the middle of that nanopore channel. So this is a great way to sequence the actual mRNA sequence. You can pull that mRNA molecule, that single molecule, through the channel and measure the actual nucleotide sequence. But if those nucleotides are modified, what it actually does is it actually impacts the electrical signal and allows us to also directly detect the actual native RNA modification. So directly detect the actual RNA modification itself. What it's sort of directly detected as is kind of a bit of a squiggle for that signal. So the signal changes, it becomes a bit erroneous. And so that allows us, that's an indication that that nucleotide is not canonical, it's got a modification. This approach, like I said, has been pretty powerful. It's been really used to pretty well characterize the modification landscape across the cell. So a lot of that epitranscriptome is being now like routinely characterized using nanopore sequencing to kind of understand how it's distributed, how those modifications are distributed and dynamically regulated in the transcriptome. There's a couple of the examples below. So if you go on one more slide. This is what we did. So obviously, like I said, we kind of focusing a little bit more on the sort of, I guess, the use of these tools for sort of therapeutic clinical development. In this case, what we did is, like I said, you can use this method to find modifications, and you can find it to find new de novo modifications. You'll find an error. And so if you have a chemical lesion, you know, a nucleotide lesion, some sort of expected analytical, like sort of chemical adjunct or something like that, it'll come up as an error. What we have to do, though, is if you want to actually correctly identify that nucleotide, what you have to essentially do is you have to retrain that base caller. And so you can see on the left-hand side, you can see there's all the errors at the different uridine, pseudouridine residues. What we did is we teamed up with the scientists at Oxford Nanopore Technologies, and we worked with them essentially to build first a training data set, so like N1 pseudouridine mRNAs, and then use that training data set to essentially train a modified aware base caller. You can see we got to, at the moment, this first generation model, it's about 93% accuracy. So, you know, there's, I think, second generation models can obviously start pushing that number up, but certainly it's fit for purpose when it comes from the perspective of like analysis and sort of, I guess, quality control applications. If you move on to the next slide. Yeah, and here's just a little bit of a kind of what the data looks like and what you'd expect to see. So at the top, this shows like an mRNA vaccine, a modified mRNA vaccine that's been sort of sequenced and analyzed using a canonical base caller. You can see that it looks pretty ugly. You know, there's lots of errors at the modified nucleotides. It's really kind of struggling with those with that sequencing. And then if you look down below, this is actually what the data looks like when you have the correct, in this case, the N1 pseudouridine aware base caller. In this case, it's cleaned up the signal a lot, and you can see that each of those kind of gray bars, that actually represents a individual sort of single mRNA molecule. And what we can do is we can use this essentially to sequence along that molecule, identify which nucleotides are modified, check that they're accurate, check that they're correctly incorporated, and check that essentially, you know, the modifications on that mRNA molecule are correct. Again, and sorry, if you add this, if you jump on one more slide. And again, it's obviously also the same principle with that 5' cap. You can use that kind of, that signal to essentially detect the modified 5' cap, other non-canonical caps as well, and essentially confirm the presence of that 5' cap at the 5' termini of your mRNA as well. And obviously this is a nice way, a nice quantitative measure, quality control measure for ensuring the capping efficiency of your sample. Next slide. Yeah, okay, cool. So look, that gives you a bit of a sense, I think, of some of the new technologies that are coming down. I mean, I know afterwards Mark's going to dive into a little bit of a deeper dive in terms of the quality control of sort of RNA modifications, and in particular the analysis using mass spec technology. So I won't go too much onto it. But I also do want to highlight that coming over the horizon pretty fast, there's a wide range of like sort of, I guess, new modifications as well. In sense, this is obviously the new 5' caps, the new modifications that I mentioned, you know, a lot of them inspired by nature, but also, you know, derived from sort of synthetic or artificial bases as well. There's also, in some ways, like a Cambrian sort of explosion in a way of different sort of mRNA modifications and topologies as well. So we're starting to see like, you know, multiple caps, multiple tails, branch tails, you know, a really bunch of different creative sort of modifications that are all really driving to kind of, I guess, extend the performance of this mRNA platform. And what does that mean? It kind of means extending the expression, improving the expression, the dosage of the mRNA, improving the stability, improving the durability of the mRNA, and typically reducing the immunogenicity or the reactogenicity. I always get them a bit confused, but reducing the reactogenicity of the mRNA as well. Next slide, which I think is my last one. Yeah, so, and one thing I do want to kind of just highlight then, I mean, as I kind of, we do sort of see these kind of new technologies that are coming over the horizon, this sort of explosion of new exciting technologies. I do think it is sort of instructive as well to take a little bit of a look at the, or sort of a bit of a history, the developmental timeline of sort of RNA modifications within the siRNA therapies as well. You know, siRNAs, you know, when they sort of initially started out, they were largely like unmodified or only had slight modifications. But over the decades, you know, over the years, we've seen like a lot of different modifications, a lot of different strategies undergo some pretty intensive preclinical and clinical research. And I think it's largely a big result of those innovations now is we're starting to see sort of siRNAs being a lot of really successful approvals, particularly in the last years, and we're starting to really see the siRNA platform, you know, start to achieve that potential. So this includes, you know, we've seen like, you know, orders of magnitudes reduction in terms of the dosages and the effectiveness of it, you know, the duration of it pushed from days out to months in terms of the actual duration of siRNAs, and also the sort of targeted selectivity. And these kind of innovations in the actual modification, the RNA modifications, look, not all of these modifications are going to be compatible with translation, are going to be, you know, compatible with mRNAs. But I do think there's quite a lot of lessons and quite a few kind of insights that we can gain from the from some learnings from the sort of the development of RNA modifications during the sort of the clinical and therapeutic development of siRNAs as well. And so, look, it's on that sort of, I guess, positive note that I'll finish up. I know my time's up as well. This is just my final thank you slide. No one's name's there, so I do apologize in terms of the acknowledgements. I mean, look, I kind of got, this is a bit more of a, in some senses, a bit more of an oversight or a bit of a review of the field that the AMM asked to do. So, you know, but I do want to kind of, I guess, in some ways, thank and acknowledge the sort of terrific team of scientists back in Australia who are really sort of developing and pushing the window a lot of on these RNA modifications and sort of developing and incorporating them in the development of these sort of new mRNA medicines as well. So I'd like to thank them. I'd also obviously like to thank you all for logging in today, logging in this morning to hear us and kind of, I guess, engage within the sort of the new trends and developments that are happening within the mRNA therapies and RNA modifications. So thanks for joining us. And also I hope you hang around for the panel discussion afterwards for some questions. Thanks.
Thank you, Tim. Our next speaker is Dr. Mark Dickman. He is at the University of Sheffield. He is he's a member of number of research centers institute at the University of Sheffield, including the Curbs Institute, Advanced Biomanufacturing Center, and the Sheffield Institute of Nucleic Acid. He's currently the Director of Student Recruitment in the Department of Chemical and Biological Engineering. He worked on scientific developing analytical techniques, including DNA, RNA chromatography, and he'll talk to us today about analysis of modified mRNA by LC-MS. Mark, the floor is yours.
Thank you very much for the introduction. Good morning or good afternoon, everyone. So yeah, I'm going to talk a little bit more focusing on the analytical aspect side of things. So really just focusing on really how we use analytical techniques, really centered on two main techniques that we use in the lab, chromatography and biological mass spectrometry, to really give us pivotal information about mRNA therapeutics. Next slide, please. Okay, so this is just a bit of a summary of the RNA analytical facility we've established at the University of Sheffield. So we have a wide range of analytical techniques that we use, focusing primarily on two main techniques, and that's the chromatography, the bioseparations, and the mass spectrometry analysis. But we also employ a wide range of orthogonal techniques, such as capillary electrophoresis, spectroscopic methods, and size exclusion and dynamic light scattering. So all these techniques, really, we focus on looking at nucleic acid therapeutics, so both mRNA vaccines and mRNA therapeutics, as well as the oligonucleotide therapeutics that Timothy just kind of talked about. Next slide, please. So within the research group, we really predominantly focus on using these two key techniques to really look at a variety of critical quality attributes of the mRNA therapeutic. So these are the key aspects or the key attributes that really kind of affect the efficacy, the safety of the mRNA therapeutic. So there we have the mass spectrometer sitting quite central there. So we develop a whole variety of different methods and apply these to look at these critical quality attributes, such as the length and heterogeneity of the poly-A tail of the mRNA, the 5' capping efficiency. We can also use it to look at accurate mRNA quantification. But what I'm going to do today is really just focus on a couple of the analytical techniques which we focus on looking at mRNA identity using direct mRNA sequencing. And this also allows us to look at the mRNA modifications within a sample. And then the other aspect is where we can simply digest the mRNA to the nucleosides look at the base composition, and also get information on the mRNA modifications that are present. Okay, next slide. Okay, so in terms of kind of introduction, I don't really need to spend too much time on this because previous talkers have really kind of introduced the topic. So within the field of the mRNA medicines, essentially we use in vitro transcription reactions to manufacture the mRNA. So essentially it's a very simple enzymatic driven procedure where we have the DNA template molecule, we have the building blocks, the NTPs, and then predominantly T7 RNA polymerase is essentially used to synthesize the mRNA from the DNA template. As we've been shown before, there's key benefits of using modified NTPs. This gives you additional stability and a reduction in the immunogenicity of the mRNA therapeutic. So primarily pseudouridine and 1-methyl pseudouridine has been used, replacing UTP in the reaction. And this generates modifications within the coding sequence. I think more recently, there's a lot more different modifications that have been employed compared to the two that originally used for the SARS-CoV-2 mRNA vaccine. So we're seeing a plethora of different modifications now being introduced. And also this adds additional complexity because many of these modifications are now not included at 100% modification. So we get lots of NTPs that are put in at 50%, 25%. So we get a range of different incorporation efficiencies as well, which complicate the analysis. So we have our particular mRNA molecule down there shown bottom right of the slide, where we have these critical quality attributes that we need to measure, such as the 5' cap, the 5' capping efficiency. And then we essentially need to confirm the identity of the mRNA sequence. Traditionally, that's done using Sanger sequencing and next-gen sequencing approaches. But I'll hopefully highlight today some of the advantages of using sort of direct RNA sequencing using mass spectrometry workflows. And this also gives us key information on the mRNA modifications that are present. So essentially, the techniques we use could be kind of broken down into two kind of key classes. One that actually gives you site-specific localization of the RNA modification. This is a technique called RNase mass mapping. And then the other technique where we don't get site-specific localization, but it's a really powerful tool to look at accuracy and quantification of the mRNA modifications that might be present on your molecule. And that's achieved through nucleoside analysis and LC-MS. Okay, next slide, please. So focusing on the first technique, and this is where we essentially take our RNA molecule of interest and we essentially degrade that using either a mixture of different enzymes or hydrolysis in acid conditions. We essentially break down the mRNA molecule into the components, into the nucleoside components. So what this is, it gives you a very powerful approach, and this has been used widely to look at biological RNAs, looking at the mRNA modifications in biological RNAs, and now it's obviously been extended to look at these mRNA vaccines and therapeutics. So here's two traces of the type of information that you get through that. So here's an example on the top trace, just mRNA synthesized using the canonical UTP. So under enzymatic hydrolysis, we essentially break the components down into the 4 main building blocks. We can simply do the analysis using UV. So you can put the standards down, look at the retention times to confirm the amounts. And you can also use the extinction coefficients also for quantitation. And then the second chromatogram shown there at the bottom, we have the mRNA that's been synthesized using 100% N1-methyl pseudo-UTP. And you see there we now have the signal intensity from the N1-methyl U. So it gives you a very simple, powerful visualization and quantification of the modifications that are present. And this type of analysis has been widely used to look at over, tens, if not hundreds of different mRNA or RNA modifications that are present on biological RNA, such as tRNA. Next slide, please. So this is a further example of where we complicate the analysis a little bit more. So as I said before, there's kind of emergence to synthesize a lot of these mRNA therapeutics where we have kind of mixed ratios. So where we replace not just the UTP with its modification, but where we may have a mixture of both 50% UTP and 51% N1-methyl UTP. And again, for this type of analysis, this approach is really powerful because we essentially separate all the potential modifications that are present in there. We can use reference standards using LC-UV, or just simply go straight into the LC-MS to identify and characterize each of those different modifications. So shown in this slide, here's an example where we've got an mRNA molecule with both 50% UTP and 51% N1 methyl U in there. And we can now see in this case, we identify both the U and the N1 methyl in there. And they're highlighted on the LC-UV chromatograms. Those of you with eagle eyes will probably show that the peak intensities aren't 50-50 like you would expect, but you do have to take into account the difference in the extinction coefficients between the two. So U has a much different extinction coefficient compared to the M1 methyl. So you need to use the peak areas in conjunction with the extinction coefficients, which then give you quantitative information. So this is a very powerful approach to look at mRNA modifications or RNA modifications, but it doesn't tell you where the modification is. It just gives you the information on the total amount present in that particular RNA molecule. Next slide, please. So if you want further information about the mRNA modifications and where they are, then we have to switch to an alternative approach. And this is essentially an approach called RNA mass mapping. So we've essentially developed this approach and applied it to look at mRNA therapeutics. So we're essentially sequencing the mRNA molecule. And the way in which we do this, we take our large RNA molecule, and then essentially what we first do is digest it with a nuclease, such as RNase T1. And in this case, cleaves after G residues on your mRNA. So we're taking these really long RNA molecules, we're chopping them up into much smaller fragments. And then what we do is we then separate those out using ion pair reverse phase chromatography. And then we do the LC-MS analysis. So we then separate all the oligo fragments that we've generated, we separate them out, and we identify them based on both their accurate mass, and also we do tandem MS. So we essentially sequence the fragments. And then with some automated software solutions, what we can do is we can take all this mass spectrometry data, we can identify the oligos and map them back to the original RNA sequence. So we're essentially doing sequence mapping, and which you may be familiar with in the way in which the protein biotherapeutics and peptide mapping has been achieved. So there are a number of caveats of this approach when we come to analyze really large RNA molecules, and I'll kind of talk you through those and also show how you can also use this same technique to confirm RNA modifications and give you identifications of where those sites of modifications are present in your mRNA molecule. Next slide, please. Okay, so this is just a schematic of the kind of the workflow, what we do. So there's your mRNA molecule with 5' cap, coding region, a large poly-A tail. So traditionally, this could be done or achieved using an RNase T1 digestion. You digest to completion, but due to the size of this and the number of Gs or the number of bases, different bases that are present, if you do a complete T1 digestion, you get a lot of what we call non-unique oligos. So they're labeled on the right-hand slide here. And these are oligonucleotides that essentially have the same sequence and will map multiple times to your mRNA molecule. So this can be a problem for sequence mapping, because really for sequence mapping, you only want unique oligos. Sequences that only map once to your mRNA molecule. Okay, so next slide, please. Next schematic. So what we've developed is to get over this problem of these non-unique oligos that are generated from complete T1 digestions or using these high-frequency enzymes, is to do a more controlled digestion. So we do a partial RNase T1 digestion. So what we simply do is we have our RNAs immobilized on magnetic beads, and we can simply incubate the reaction for a defined period of time. And we can quickly stop the reaction by taking the beads out. So this allows us to do a very controlled reaction. We can stop the reaction, say, after 10 minutes. So what this generates is much larger fragments, and it gets away from these non-unique oligos, which map multiple times across your mRNA molecule. Next slide, please. Okay, so this is a bit of an example of using this particular workflow, some of the data that you get. So here's a couple of examples where we've taken two different mRNA sequences that we've manufactured and purified in the lab. So there's a SARS-CoV-2 spike mRNA and an EGFP reporter mRNA. So if we do our partial T1 digest using the strategy or the workflow that I've just demonstrated, you see the chromatograms on the right-hand slide. We essentially generate a large complex series of oligo fragments, where each one of these peak is essentially a single oligonucleotide, or a single might contain one or two different oligonucleotides. So we get a complex set of different oligo fragments. And then essentially, as these go into the mass spec, we essentially determine the accurate mass of these, and we use tandem MS to essentially sequence those and match them against the original sequence. Okay, next slide, please. So when we do this workflow, we get very good reproducibility in the digestion. So here's this example for EGFP mRNA, where we did 3 different digestions under exactly the same conditions. And you see we get very reproducible chromatograms. The digest is very reproducible. And you can easily control that as well. You could either change the time, change the temperature, change the amount of beads to kind of control how you do the different digest. And then we get very high resolution accurate mass. So all our analysis is essentially done on high-end Orbitrap tandem mass spectrometers, which gives us really high mass accuracy. And we get very good sensitivity. And here's a couple of examples of 3 different oligo fragments that we've identified. We get very good retention time stability across the different replicate experiments. Okay, next slide, please. So this is what some of the data kind of works like that we get from the mass spectrometry data. So here's an example on your left-hand side of the screen. We've got a particular oligonucleotide sequence there. And we'll get a different number of different charge states for that. So here we get the charge states of 13, 12 for that particular oligonucleotide. So we get the high resolution mass spec data, but that doesn't tell us what the sequence is. So even though we've got high resolution accurate mass for that particular oligonucleotide fragment, it doesn't tell us what the sequence is. So to do that, we have to get the tandem MS information. So this is where we essentially fragment that oligo fragment. We basically hit the oligo fragment against an inert gas, and that breaks it apart into a series of well-defined different ions. And that's showed on the bottom of the screen there. So we get lots of different ions that we generate, and each of those essentially can be mapped back and gives us essentially the sequence of that particular oligonucleotide fragment. So on the right-hand side of the slide, I can show you, essentially shows you how powerful this tool is. So here now in an example, we've got 2 different sequence isomers. So these actual oligonucleotide sequences have exactly the same mass, but you can see they have different sequences. And if we do the tandem mass spec analysis, we essentially can get the information from the different sequences and identify those. So although they have exactly the same mass, the sequencing information allows us to discriminate those and identify the 2 different sequence isomers that we generate. Okay, next slide, please. So this is the workflow. Essentially, we've also, the automated analysis is really important. So historically, we did a lot of this workflow kind of in-house or using an in-house software where we would have only a small RNA fragment or a small RNA species. It would only generate 5 or 10 different fragments. And we kind of would do manual assignments of those. But obviously, when it comes to analyzing these really large mRNA therapeutics, you know, they're up 4,000, up to 10,000 nucleotides in length, we generate thousands of different oligonucleotide sequences. So it's really important we now have automated data analysis tools to do the identifications for us and map these back to the original sequence. So the approaches we've taken, working collaboratively with our colleagues in Thermo Fisher, is to essentially use Biopharma Finder, which is an automated data software analysis. So it essentially takes the data that we generated in the mass spectrometer, and we have an automated pipeline now of how we do the sequence mapping. So essentially, the first part of this is just putting in your sequence. Okay, so all you need to do is put in your mRNA sequence of interest. Then you essentially just select the RNAs that you're going to use for your digestion, such as RNase A, T1, Mu2, etc. And then what it does, it analyzes the data, searches all the information, all the sequence information you got against the theoretical sequences that are likely to be generated from that particular fragment. And then it essentially matches and identifies all those there shown in red. And at the end, it generates a sequence coverage map. So it shows you which oligo fragments you've identified against the original RNA sequence. And then it gives you an overall percentage coverage that you managed to obtain. Okay, next slide, please. So for the analysis of these large mRNA therapeutics, what we did was a comparison of our new workflow where we generated this partial T1 digest strategy, and we compared it against the traditional complete T1 digest. And we looked at the sequence coverage that we got for 2 different mRNAs, the EGFP mRNA and the SARS-CoV-2 spike mRNA. So you can see here we've got clear, significant increase in the sequence coverage that we achieve using our partial T1 digest strategy, which from a single analysis, this is just one sample, one single analysis in the mass spectrometer, we can achieve over about 85% sequence coverage. Whereas if you use the complete T1 digest, not using those non-unique oligos that map multiple times. If we remove those, then we only typically achieve below 25% sequence coverage. Of course, for any mass spectrometry workflow or any analysis, it's really important to ensure that we're getting accuracy and robustness. So what we do for the mass spectrometry workflows is when we do the analysis, we put in a random sequence of exactly the same size, same GC content, and we search our mass spectrometry against that random sequence just to make sure we're getting no significant hits or matches to that random sequence. And here you can see for these two examples and the data, we get less than 1% matches against that particular random sequence. Okay, next slide, please. And this is the resulting kind of sequence maps you get at the end of that. So you can see here, single analysis, we do get the occasional gaps in there, and they can be filled in from replicate experiments, or using different enzymes, or changing the digest conditions. So it's a fast workflow, it's direct, so we work very closely with Zoltan Kiss's team at the University of Sheffield, who do a lot of mRNA manufacturing of therapeutics. So from the time taken, and then they give us an mRNA sample within less than 90 minutes. We can do the digest, do the analysis, and generate the sequence mapping in the automated data analysis. So we can do essentially mRNA identity testing within less than 90 minutes, generating typically 90% sequence coverage. So I'll just highlight some. This was a recent publication that we published in Analytical Chemistry just a couple of years ago. Okay, next slide, please. Okay, from an aspect of looking at mRNA modifications, it's really important, obviously, this workflow is able to identify the modifications that are present. So it's really simple to do that. So this occurs from when you put your sequence in to the software, then essentially putting a sequence in with all the, say, the U's in the sequence, then you essentially just replace that with your modification of choice. So you can simply just, when you add the sequence to the software, you just put a sequence in where all the U's are replaced by N1-methyl pseudouridine. We'll talk about pseudouridine a little bit later because that's a little bit more challenging because that's a mass silent modification. But essentially, you can just add the modification of choice, hundreds of different modifications you can choose from, simply just put into the software. And then essentially the mass spectrometry data essentially searches this mRNA sequence now with the modifications present. Next slide, please. So here's some data where we've got some data looking at both the unmodified and the modified mRNAs. So here's an example just on the left-hand slide. We've got the FLUC mRNA that contains the UTP non-modified versus 5-methoxy-U. And you can see for both the analysis, essentially all we do is we change what sequence we map against. And in both cases, we get virtually identical sequence coverage, over greater than 95%, 90%. And then if we delve into the analysis in a little bit more detail, so what I've shown you on the right-hand slide now is where we really validate the presence of the modifications. So I've identified, or I've got the mass spectrometry data showing the exact same oligonucleotide sequence that we've identified, where we've highlighted in red where the U's, in this case the 5-methoxy-U's, are present now compared to the non-modified in the above strand. So the way in which we do that, we get a series of different ions that I've highlighted in red. So these are specific ions with masses that are different to the non-modified strand. So this tells us where they are essentially present. So by looking at the different ions, fragment ions that are present, this gives us or validates the site-specific incorporation of these modified U's in that particular sequence. So it's really powerful. It's really doesn't really depend on the mass. We can put any modification in as long as it's got a mass difference. Then we'll see the mass difference both at the level of the oligonucleotide and the fragmented oligonucleotide. So it gives us the site-specific modification. So here we are on that right-hand side, same oligonucleotide. We use the mass spectrometry to confirm that we've got, in this case, we've got 4 different modified U's present in that strand. And we've got the mass spectrometry data to confirm that. Next slide, please. So that was the example for 5-methoxy-U. We've got works exactly the same for N1-methyl-U. I should say when it comes to pseudouridine, this is a little bit more challenging when it comes to mass spec analysis. This is essentially because it's mass silent. So any modification is essentially isobaric, has the same mass. However, there's a number of strategies you can use mass spectrometry to get around that particular challenge. So I've just highlighted 3 here. The references are below from the different groups that have essentially employed these strategies. So what you can do if you've got pseudouridine in there, then a number of groups have chemically derivatized that. where that modifies the pseudouridine, such as acrylonitrile modifications or using methylvinyl sulfone modifications. And that will site-specifically modify only the pseudouridines and not the uridines that are present. A number of groups have used in vivo labeling approaches. So this has really been focused on the biological RNAs. So if there's pseudouridine in the tRNAs or ribosomal RNAs, then you can use deuterium labeling, which gives you powerful stable isotope labeling approaches. But probably one of the easiest approaches is looking at the diagnostic ions that are present in that tandem mass spec data that I showed you. So here on the left-hand side, if you look at the tandem MS data, you get diagnostic ions or certain masses that are only present from pseudouridine and not the uridine. And these are these ions at 207 and 164. So if you look very closely at the tandem mass spec data, that gives you diagnostic ions that are only generated from pseudouridine, not uridine. So again, we can use mass spectrometry to really validate site-specific incorporation. It's a little bit more challenging because it's mass silent, but we have these diagnostic ions or alternative strategies that are quite useful. Next slide, please. The approach we take often is a bit of a combination of both. So if you're using pseudouridine, then we kind of do a little bit of a combination of both. So we use the enzymatic hydrolysis approach just to validate that we've got pseudouridine in our mRNA of interest, not uridine. And then when we do the RNA digests or the RNA mapping approaches, then we've already confirmed that we've got no U present. All our U's have been replaced by pseudo U's. So we can use essentially the direct sequence mapping just to validate or give us the site-specific information where all the pseudo U's are present. Next slide, please. Okay, with that, I'd just like to finish and just really acknowledge some of the people in the group that have done a lot of the work. So in particular, the direct sequence mapping approach has largely been done by Christina and Emma in the group. A lot of the work with the enzymatic hydrolysis has been done by Caroline Evans in the group. We work very closely, as I said, with Zoltan Kiss's team here at the University of Sheffield that did a lot of the manufacturing of the mRNA molecules that we've worked on. So thanks to Adi and Zoltan and his team. And also thanks to our colleagues at Thermo Fisher, who we've worked closely with in terms of developing the direct sequence mapping workflows and some of the software tools. And in particular, finally, I'd just like to thank our funders at UKRI and welcome me.
Thank you, Dr. Dickman. Appreciate it. And now, I'd ask all the speakers to join me for a few Q&A before we close this webinar. The first question I have is, I think for Dr. Hugh, what do you see the future of RNA modification in medicine?
I think there are a number of possibilities. I think obviously mRNA vaccine is one way to move forward, but I think both messenger RNA methylation, but also chromatin associated RNA methylation have plenty potentials. As I said, for cell therapy, for targeted therapy, because we really uncover a new regulatory pathways, and whenever we discover a new regulatory pathway, there are always interesting applications to help solve clinical questions. So I think the field is broad, and we just need to look at a specific case.
Tim or Mark, do you have anything to add to that? Okay. The other question is, have there any studies to support multi-modification in the same mRNA? So one that you have, N-methyl pseudo-U and a 5C, or multiple ones, and does that have any impact on translation?
I could take. Yeah, go ahead, Tim.
You go.
Go ahead, Tim. Go ahead, Tim.
Okay, I mean, look, certainly, so there's two aspects. I mean, certainly in some senses, like an mRNA has lots of modifications because obviously got the cap and the pseudouridine and stuff like that. But certainly in terms of like mixing different modifications within one mRNA, I mean, I haven't seen it in terms of clinical, like in terms of clinical research, but certainly within preclinical research, it's been it's pretty well investigated, and it's also pretty common as well. I mean, we talk about sort of, let's say, N1 pseudouridine as in substituting all the canonical uridines for the pseudouridine. You can also put in like a mixture, so you could put in 10%, and therefore, like randomly, about 10% of your nucleotides will have that modification as well. So it's possible to kind of, I guess, mix and match, and also to like change in some ways the number of modifications, the fraction of nucleotides that are modified within your mRNA. But I haven't seen, like, I guess, in some sort of senses, multiple modifications being used together in concert.
Yeah, just to add, I think as Tim said, there's quite a lot of preclinical work on using lots of different modifications and different ratios, etc. Some really nice recent work using 5-methyl-C combinations, lot nucleic acids, that kind of thing. I think just one kind of caveat there from an analytical perspective, this makes it very challenging to analyze. If you mix something in at 10% modification, you don't get 10% at every site. So you open up you're making an mRNA molecule with lots of different heterogeneous population now. So this gives a big analytical challenge, because you're generating a bigger pool of different population of species. Do you have to characterize those? What are the regulatory requirements going to be moving forward? It makes it a significant analytical challenge.
Okay. Doctor, you have something to add to that?
Not really. I think that's a very good summary, but I just want to say on native transcript, there are indeed multiple modifications, and still, I think a research subject whether these modifications have any synergistic roles, we do not know at this moment.
Okay. So there's still a lot to be done in that area, because that could be in a positive way or a negative way to have multi-modification to mimic nature, but it'll be synthetic. So we have ways to go on that. Tim, you've showed some work on the nanopore. It's really interesting, and this is really catching up quite a bit and becoming a technique that a lot of people trying to adapt. Some of the work that you've done, have you done multi modification on the same mRNA using the third generation sequencing?
Yeah, we've done a handful of different modifications, certainly. We've only trained in terms of training a base caller, that's only really for the pseudouridine from that perspective.
Okay.
But certainly there is also, I mean, I'd actually say if you look in the literature, I mean, and this is going back as well, that kind of within the cell, there's actually been a lot of people who have developed methods already to detect or identify specific modifications. Like I said, in the case of nanopore, that can be basically trying to identify that electrical signature that's specific for the modification. But there's also other techniques as well. So what you can do is you can take your RNA, you can convert it chemically, you can put modifications to it such that when you sequence it, you can actually detect where those mutations and so forth are using like next generation sequencing. So certainly, I mean, the example I used was pseudouridine, but certainly you could pick up many other different diversity of modifications using the same approach.
And would you be able to pick up if you have a mixture of uridine and pseudouridine?
Yeah, you could. So you could do it on a base by base level. Essentially, what you'd be calling it on a per nucleotide level. So this one's canonical uridine. The next one is a pseudouridine. The next one's canonical. You could certainly distinguish between those two signatures, and do it on a base by base level. Yeah, if you're not at that 100% level.
Okay.
Mm-hmm.
I mean, Dr. Hu, sorry.
I was just going to say, we discussed this for a lot of the challenge, I think, is probably more in the bioinformatics and the informatics rather than technological. So it's really kind of training that base caller, analyzing it, and getting it accurate. That's where a lot of the challenge remains.
So this is going to dovetail a little bit more into my next question is, Mark, you've showed some really great data, and something that I've done in the, kind of similar to what you've done, using two techniques to identify modified mRNA by, base analysis and LC-MS fingerprinting. And the LCMS fingerprinting is also heavy on the bioinformatic. Have you thought or have you done any work that to kind of bridge the sequencing and the LCMS fingerprinting, as an orthogonal to show that the data is comparable?
So we've done a little bit of work. So I think one of the issues is, to a certain degree, that if you go down the sequencing route, then a lot of the methods are potentially indirect methods. So you really want to be comparing how Tim's nanopore approaches, where you are doing the direct sequencing type approaches. So you really are kind of more comparing apples with apples. So I think one of the problems of the kind of traditional sequencing approaches is that they are indirect methods, where you're converting the RNA into cDNA. So I think if you are going to do a kind of a side-by-side and kind of an analysis, then you ensure that you are using a comparative, direct sequencing approach versus similar to the mass spec workflow. And I kind of think there's advantages and disadvantages of both to a certain degree. So I think from my aspect, from what the way we look at it is, you should be always using orthogonal approaches for each of the critical quality attributes. So I would kind of say there's, in this case, using the direct nanopore, we see it as an orthogonal method to the mass spectrometry workflow, each with its own kind of advantages and disadvantages.
Okay, thank you. One other question is, what's the most important aspect of post-transcriptional regulation through RNA methylation? I think, Chang, if you can take that question for us.
Can you say it again? Sorry, I missed the first word.
What is the most important aspect of post-transcriptional regulation through RNA methylation?
Right, I think that's stability. It's pretty clear that we had this paper published, I think, last year, showing that in mammals, or actually in animals, there's a co-evolution of exon architecture, or the length of the exon, versus RNA M6A methylation. And that co-evolution regulation really dictates transcript stability. So essentially, from fish to mouse to monkey to human, there's this control of exon length, which controls transcript stability through mRNA M6A methylation. But beyond that, there's also translation regulation, export from nuclear and cellular localization regulation. But I would say stability is the most important function.
Tim, Mark, anything to add?
No, I mean, I think, it was really impressive seeing that sort of like the transcriptome and the clearance and that sort of side of things. Although I do think, if we're talking about my favorite applications or my favorite phenomena that's based on RNA editing, I do think, I know the octopus or the cephalopods, their long-term memory is actually based on RNA editing, RNA modifications. So their long-term memory, their synaptic plasticity, unlike, they've got a different type of intelligence to us, and their one is actually nested in RNA modifications. So I think it's sort of a fantastic sort of, I guess, indication of what RNA modifications can do in the natural world.
One question came up is, I know this was done, but this most likely for you, Tim, is, have you with nanopore sequencing, have you been able to accurately determine the poly-A length or poly-A characterization?
Yeah, they do. So they do. So poly-A tail length, you can accurately determine. There is a bit of software out there called Tail Finder, which does it, like a bit of off-the-shelf academic software. And I believe they are building it into the EPI2 ME workflow at the moment. But yeah, the short answer is it is actually pretty accurate. I was pretty surprised. It is actually pretty accurate for measuring that poly-A tail. And since we're talking about RNA modifications, I think they've also got, because now there is a bit of sort of fashion in terms of actually modifying the poly-A tail as well, putting like uridine residues, these sorts of things on at the end as well. And so I think it also is compatible with starting to pull in some of those modifications or non-template additions as well.
Okay.
But the short answer is yes.
Well, I would like to thank all our speakers. I think this has been a very wonderful webinar with a lot of valuable information. So we thank you all, and thanks everyone for calling in and joining us on the webinar today. And with that, we conclude our webinar for the day. Thank you, everyone.
Fantastic. Thank you.
Thank you.
Cheers.